Good afternoon. I'm going to talk about ancestral origins of MJD and the Joseph and Machado lineages, their sublineages spread worldwide. And I'd like to start by acknowledging all the authors in this collaborative study. As you can see, this is an extensive list and many of you are in there. A collaborative study like this wouldn't be possible without your contribution. Thank you. So as you know, the expansions responsible for MJD, they occurred most likely from the normal range of alleles at least twice. And because this is a dynamic mutation, so in a few generations, we end up with a high level of polymorphism in the expanded range of alleles. And this wouldn't be a problem to assess mutations history if we had only a single de novo expansion. But in MJD, there is more than one mutational event. So let's say that we have here the first one, and then diversity accumulates due to the intergenerational instability of the CAG expanded alleles. And then, let's say, a second mutational event occurs and more diversity accumulates. So this, it's a problem because if we analyze only the CAG repeat, we are going to look at different evolutionary histories overlapped. So to answer questions like where did these mutations occur? I mean, their populations of birth, how long ago, and why? I mean, the genetic background, because we didn't know if there were predisposing haplotypes or not. So to answer all these questions, we need to analyze the flanking regions of the CAG repeat. And we did with the polymorphic sites, both SNPs and STRs, to capture variation from different time scales because they have different mutation rates. And finally, because we wanted to assess the background segregating with expanded alleles, not normal chromosomes, so we inferred the expanded haplotypes exclusively. So previously, we have shown that the worldwide spread mutation is of Asian origin based on the high diversity level found in the Japanese MJD families compared to other MJD populations. And actually, we named this Joseph lineage since this haplotype is present also in Flores, the Azorean island home of Joseph family. And on the other hand, the same way we named the Machado lineage because this haplotype is very frequent in San Miguel Island, so where this Machado family is from. But back then, we studied only in the beginning 3 and then 6 SNPs in this study. So we wanted to expand this haplotype study and to see if additional markers could distinguish more MJD haplotypes. So we are currently analyzing a set of 30 SNPs spread over 15 KB regions around the CAG. And up to now, we have completed this analysis for 370 MJD families from 30 populations. We sequenced and then aligned with the Genius software. As for the fast-evolving markers, we selected 7 STRs, and flanking the Ataxin-3 gene. And we genotyped them in a multiplex reaction, actually in an octaplex, because we included also the CAG repeat. Here you can see the normal allele and the expanded allele. And then we inferred haplotypes, disease-associated haplotypes by segregation and sometimes bioinformatically with phase. Phylogenetic networks were done with the network and the POP3 softwares. So we observed a new sublineage in Aboriginal families of Groote Eylandt and Irkalla, and this haplotype was also present also in some Asian populations and the United States. And this Groote sublineage differs from the Joseph lineage only at this SNP. And previously, when we were aware of the existence of only 11 families carrying these 11 families carrying this Joseph-derived haplotype, so we suggested that a recurrent mutation occurred in this SNP via Asia, because we found the haplotypes from these families phylogenetically closer to the Japanese families when compared to other populations. And also we suggested that the introduction of MJD expanded alleles in these communities was probably via Asia. So now we expanded this study, and we have observed this haplotype in 36 additional families. Also, again, in the same populations, mostly Asia. And based on the high diversity that you can observe here, let's say each circle represents a different STR haplotype, and the length of the lines is proportional to the number of mutations, of STR mutations between different STR haplotypes. So looking at this diversity, we think that the origin of this sublineage occurred shortly after the Joseph origin, the origin of the Joseph lineage. So another background we found in families from Africa, Spain, United States, and the Yemenite Jewish families presented before lunch by Professor Gordon. So, and because we found diversity in African families and a founder haplotype among these Yemenite Jewish, we think the introduction in these families must have been recently, likely from a common ancestor of African origin. As for the Machado lineage, it's restricted to Iberia and United States, and we explain the high disease prevalence in Portugal by a founder effect, as here observed, because the circle size is proportional to the number of families. So all these families share the same STR-based haplotype. We also observed a Machado-derived lineage in a few families. Actually, two of them described yesterday by Dr. Mario, Peruvian families. And actually, I was surprised because when I heard that at least one of them is of Asian origin. We are going to explore this further later. And so, to conclude, we found the highest gene diversity in Joseph lineage families from the United States, Asia, followed by European populations, Portuguese families showing the lowest. And we are still trying to understand this new mixed haplotype that we found in the family from French Guiana, because this family has a core haplotype similar to the Joseph lineage, but then a flanking haplotype more similar to the Machado one. So we are still trying to understand it. To wrap up, we can say that MJD causing expansions do not seem to occur frequently on multiple background haplotypes, and MJD prevalence may be explained mainly by migration rather than de novo events. And finally, a huge thanks to all patients, families, and the communities. And from my groups in Porto, led by António Mourinho and Jorge Sequeiros, And I would like also to thank the organizing committee for the opportunity to present in this meeting and for this amazing meeting. Thank you.
Thank you, Dr. Martin. That was a very interesting talk. Do we have any questions? No questions? People are feeling a little bit sleepy. Yes.
Congratulations. Such a great presentation. Just wondering, which gonna be like the best strategy to try to understand the case, for example, for the Peruvian families. As I mentioned before, as far as I remember, that 2 families were sent to court on these things. One of them had a clear affected ancestor originated from the UK, and the other one was the one what we were talking about, Trinidad and Tobago, an affected ancestor as far as we got information from families. It will be kind of interesting to see what is the haplotype composition of the other 3 families as well.
Yes, exactly. Yeah. I'd like to complete. Yeah, I'd like to complete this study. Thanks.
I have a question. For the families that were from Mali and Guyana, how were you able to genotype those individuals? Were they somewhere in another country, or were you able to access them within those places?
Well, many samples were sent to Jorge Sequeiros lab, so we have a collaborative study. Very often we received DNA samples from other clinicians or researchers.
Do you have any other questions?
You mentioned in your introduction that there may be several mutations or expansions occurring. So based on your data, do we really have just two founder effects, or what would you estimate how many like original MJD SCA3 mutations did occur in the past?
Do you mean 2 major de novo expansions? Yeah, actually, we are not sure that, for instance, this Groot sublineage is really a Joseph sublineage, or if it might be a new background because of the diversity that we observed among these families. We are not 100% sure.
Okay, thank you. We'll move on to the next